Good morning, students. So, today we have to start with the experiment to show that oxygen is used in the process of respiration. Okay, and which type of respiration? That is aerobic respiration. So, in aerobic respiration, the oxygen is used. What are the things required for this experiment is we need a stand, we need two conical flasks, and we will mark it as A and B. Okay, two conical flasks we will take and we will mark these two conical flasks as A and B. We will take one beaker, we will take two delivery tube that is also attached to one A and the other one to the B. Then two test tube we need germinating bean seeds, and we also need boiled bean seeds. So why germinating bean seeds? So germinating bean seeds will perform the process of respiration, and the boiled bean seed is dead, so it will not perform the process of the respiration. Okay. Then the other thing we need is we need a wet cotton, we need soda lime, and soda lime is the mixture of sodium hydroxide and slaked lime. Soda lime is the mixture of sodium hydroxide and slaked lime. And what is the use of this soda lime? To absorb carbon dioxide. Okay, we also need water, plus we also need water will be filled in the beaker, and we also need an antiseptic. So why we need antiseptic? So this antiseptic will prevent the growth of bacteria in the second flask. Okay, so these are the things we need for this experiment. Okay, now these things will be arranged as shown in the figure. So here we have taken two conical flasks, that is conical flask A and B. Okay, we have taken two conical flask A and B. Then we have taken the stand through which these conical flask is attached. Okay, we have taken a beaker. This beaker is filled with water. Okay, we have taken two test tubes, and these test tubes are filled with soda lime, and soda lime is the mixture of what? Sodium hydroxide and slaked lime. Soda lime is the mixture of sodium hydroxide and slaked lime. And what is the use of soda lime? To absorb carbon dioxide. Okay, so whatever will be the air trapped in this flask, and in this flask, from this air the carbon dioxide will be absorbed in the soda lime. Here also the carbon dioxide will be absorbed in the soda lime. Okay, we have taken wet cotton at the base of the conical flask, and in the previous class you have studied about the germination, and in germination you know that water is essential for germination. Here in conical flask A we have taken the germinating seeds, and in conical flask B we have taken the dead bean seeds. Okay, so when there is germinating bean seeds, so these germinating bean seeds will require oxygen, and oxygen is present in this trapped air, so this oxygen will be taken up by the germinating seeds for the process of aerobic respiration. Whereas here dead bean seeds is coated with antiseptic. And why it is coated with antiseptic? So that the bacteria will not grow on it, and if the bacteria is not growing over there, the oxygen or present in this air will remain same. So the percentage of oxygen in the air trapped inside the conical flask B will not be utilized. Okay, so what is the observation in this experiment? Here the beaker is filled with what? Water. So on the surface of the water, the atmospheric air is applying a pressure, for example P. Okay, and the pressure inside this conical flask, for example, it is P1. Okay, so first observation is what? There is rise in the level of water in delivery tube A. these two are the delivery tube. One delivery tube is attached with the conical flask A, another delivery tube is attached with the conical flask B. Okay, so in which case you observe the higher increase in the level of water, that is in case A, and in case B there is only slight increase in the level of water. Now you see the reason behind it. Okay, for example, this one is P1. In the conical flask A, carbon dioxide present in the trapped air is utilized by soda lime, is absorbed by the soda lime, and oxygen is utilized by the germinating seeds. So the volume inside this conical flask is same, but the quantity of air gets decreases. And why the quantity of air gets decreases? Because oxygen is utilized here, carbon dioxide is absorbed here. That is why there will be a decrease in the pressure, and this decrease in the pressure is up to P1, whereas the atmosphere is still applying the same pressure. So a difference in the pressure is created, and when the difference in the pressure is created, this is at higher pressure and this is at lower pressure. So the water will move from high pressure towards the low pressure. Whereas in case B, only carbon dioxide, and carbon dioxide is only forming 0.03% of the air. So this carbon dioxide is only being absorbed, oxygen is not being absorbed over here. So that is why there is no sufficient, or you can say not a proper decrease, or such decrease in the pressure is negligible, and due to the negligible decrease in the pressure in this, the water will not rise over here. Okay, understood it? So here are some questions. Observation. So what is the observation? Level of water will rise in delivery tube A. Reason: soda lime absorbs carbon dioxide from the trapped air inside the conical flask. Germinating seeds utilize oxygen for respiration. So the pressure of air inside the conical flask becomes less than the atmospheric pressure. Okay, therefore the water moves from higher pressure to lower pressure. And where was the higher pressure? The atmospheric pressure is higher pressure, and the pressure inside this conical flask is at lower pressure. Okay, then another observation: there is slight increase in the level of water in delivery tube B. Reason: only carbon dioxide is absorbed by soda lime. Boiled bean seed does not respire, so negligible difference of air pressure is formed, and when negligible difference in the air pressure is found, therefore negligible rise in the level of water in delivery tube B. Now what is the use of antiseptic in flask B? So antiseptic will prevent the bacterial decomposition of the boiled bean seed. Therefore the trapped air oxygen will not be utilized by the bacteria, and therefore the flask B will act as a control setup. Okay. Now what is the difference between the experimental setup and control setup? So experimental setup will have all the conditions under experimental study is present in it, whereas in the control setup under experimental study is missing. So what is the experiment over here? To show that oxygen is used in the process of respiration, and respiration is not performed in which flask? That is flask B. So flask B is acting here as control setup, whereas flask A will act here as experimental setup. This is all for today. Thank you everyone.